She has been not just active on campus as a teacher and mentor and scholar. She's been very active as president of a campus of the University System of Maryland. She's been very active on the national international scene. I could spend an hour going over her CV with you, but I won't. But I'll just tell you that she was the first woman director of the National Science Foundation. She is a member of the National Academies of Science. She is a winner of the recipient of the Presidential Medal of Science. She's been given 62 or so honorary degrees. She was president of the AAAS, and it goes on and on and on. These honors she's received are truly well-deserved for her extraordinary contributions as a scholar. So I'm going to just leave it at that, and with a real pleasure, introducing my colleague and my friend, Rita Colwell.
Thank you so much, Art. Indeed, a very dear friend. We go back many years. I also see Marilyn Rippon, another very dear friend, who's on. Thanks so much. I'm delighted to have a chance to talk to you about what I've worked on for the last 40 years. Really, it deals with epidemics. I wanted to make sure you all, I'm sure you know, but just to recount the fact that this epidemic is not novel in the sense that we've gone through epidemics for centuries, all the way back to the plague, at least in recorded time, and very likely before that. So we're currently in actually two epidemics. We don't talk about the cholera epidemic because it doesn't have as much influence on our lives as the COVID. But cholera is pretty serious in the sense that it's a diarrheal disease, and it's the predominant diarrheal disease. It kills almost 2 million people every year. And we have billions of cases every year of diarrheal diseases, of which cholera is dominant. But as you can see, by looking at water-related diseases, this accounts for a huge mortality and a very, very high morbidity in the world. Cholera is global. It's a water-related diarrheal disease. We're in a pandemic because we have been recording pandemics of cholera, waves of cholera over the centuries. And the seventh pandemic started in the 1960s and it continues. It affects about 50 countries, about 7 million people every year. We've unfairly referred to the Bengal Delta, Bangladesh and India, as the home of cholera, but in fact, 150 years ago, Washington, D.C., Savannah, Georgia, the Gulf of Mexico, even as far north as Canada, there were cholera epidemics every summer. Those of you who hike the C&O Canal, the Monocacy Aqueduct has a plaque that is dedicated to the cholera, to the workers who built the canal and the overpass who died of cholera. In the US and in the Washington area, the workers would flee in August and September because that was the diarrhea time, the cholera time. But cholera exists in the aquatic environment. It's naturally occurring. And in fact, that's the discovery that I and my lab, my students made at Maryland, was showing and proving that the cholera bacteria are not just person to person, but exist in the aquatic environment. And that it's unlikely we can eradicate cholera because the bacteria also carry out nitrogen transfer and carbon cycling in the environment. And so the burden of cholera during the past 30 years, you can see that it's predominantly in Africa, India. In 1975, you see that countries in Africa and in Asia were afflicted with cholera. The interesting thing is that about the late 80s, there was a massive epidemic of cholera that occurred in Latin America. It was considered to be related to the El Nino phenomenon. As you can see, in 1991, South America really became hit hard by cholera. And it was a very unusual preceding years when the Pacific current that came up along the coast of South America was extremely warm, much warmer than usual. And we consider that as one of the contributing factors. And in fact, the burden of cholera is very much climate related. And I'll focus on that in the talk. Let me move on to just describe the causative agent as a bacterium. In the upper right, you can see the kind of curved rod, which is why it's called Vibrio. It was named by Robert Koch back in 1890 because of its curved shape and its single tail. And it's easy to grow. You can see the petri dish with the colonies that are bright yellow in a medium that turns yellow because of the capacity of the bacteria to use the sugars that are in the medium. And then a typical patient, even today in Bangladesh, the loss of fluid is measured because the amount lost by vomiting and diarrhea, if it becomes really severe and not replenished, the victim will die. Now, the work we did in the Chesapeake Bay, you can see the stations numbered right along the backbone of the bay. From about 1965, actually I started at Maryland in 1973. So from 1973 to the present, I still have a student who goes out and does sampling. We took water samples up and down the bay, and we made the discovery that the bacteria that cause cholera exist in the Chesapeake Bay. Now, I tell you, this was not a happy occasion for me. This was about 1975. It was particularly unpleasant because the medical community had as its mantra that the bacteria that cause cholera transferred person to person. Well, what we discovered in 1973, 74, and 75 was that the Vibrios are in the bay and they're associated with plankton, the copepod, and that relationship is quite critical. In fact, shown here is a gravid female, that is a female copepod, ready to cast her eggs into the water column. And coated on the egg sac are the bacteria because they have a powerful enzyme that breaks down the protein of the egg sac and releases the eggs into the water column. So it's pretty much probably a symbiotic relationship. In addition, the bacteria, the Vibrios, all have an enzyme, chitinase, which breaks down the hard white shell of crabs, shrimp, and copepods. And so we developed our very first model way back in 1975, 1980, that said, look, there's a relationship between the bacteria, the plankton, and the disease in humans. But in Chesapeake Bay, we don't have cholera. We haven't had it since about maybe 1930. But we do have other Vibrios, one of which is very pathogenic, Vibrio vulnificus. That is a bacterium that if you're swimming in the bay and you have an open cut or an abrasion, becomes infected. If it goes systemic, the fatality rate is about 50%. You don't mess with Vibrio vulnificus. You get antibiotic treatment right away. In fact, I lecture regularly on the Eastern Shore, and there's always a very large audience, and 3 or 4 people, one chap in a wheelchair who lost his leg had to be amputated from the infection with Vibrio vulnificus, and 2 or 3 others who've lost a finger or a thumb from that infection. There's also Vibrio perihemolyticus associated with shellfish, but if shellfish is cooked properly, it's not a problem. In any case, the other discovery we made, which was very important, was the reason the physicians couldn't ascribe this bacterium to the human disease is because you see the yellow columns. That's the number of bacteria in a given sample that you could grow in the lab. But when we developed a technique for being able to look under the electron microscope or to stain and look under a fluorescent microscope, we could see the bacteria that could not be grown. Down at the bottom right, you can see the outline because this fluorescent antibody that we linked to a molecule that glowed under UV light, you could see the bacteria that you couldn't grow. And indeed, the bottom left shows how the bacteria, when in very cold water, will turn into rounded up cells and they will become dormant. And then they will break open and start growing again when the temperature gets warm and the nutrients are present. So this gives us a global view of this disease, and it's the fact that it is a bacterium that exists in the world oceans, particularly in the coastal waters associated with plankton. And the early studies were done predominantly in the Far East, in Asia, and that is how we've come to assume that cholera is associated with Bangladesh. And that is where I've done a lot of work over the past 40 years in a locale known as Matlab, which is a bit southeast of Dhaka, the major city. And I have done most of the work that I will describe here in Bangladesh, in these villages. We were criticized severely for saying that we had cholera in Chesapeake Bay. In fact, by doing genomics, we could prove that it was, in fact, in Chesapeake Bay. But then we were criticized and said, well, maybe that's an unusual situation, but it doesn't hold in Bangladesh. So that was the challenge. And in 1975, my students and I went to Bangladesh and started work and have gone there every year, 2 or 3 times a year until, of course, COVID hit. And we found that this is where the villagers take their drinking water, and it's also where they do their personal toothbrushing, washing utensils, and collecting water for drinking. And indeed, the Bangladesh model fit what we discovered in Chesapeake Bay. The bacteria are associated with plankton. And of course, the sanitation is very poor, and clearly person-to-person transmission occurs, but the source is really the environment. Now, we've also learned that there are environmental parameters, that is, weather, temperature, salinity of the water, storms, all associated with the massive epidemics that occur twice a year in areas like Bangladesh. And so we have worked on developing a model for prediction. Why should we do that? Well, if we can intervene, as you see on the left, early, we can reduce the number of cases, the white line. But if we wait until the disease is at its peak, then intervention is sort of after, closing the barn door after the bacterium has disseminated in the community. So in our work in Bangladesh, we made a discovery. We made the discovery that in 1980, Landsat satellite was launched. And in 1985, I had made this correlation of plankton. We had really worked it out extremely well. And it occurred to me that we could use satellite sensing to predict the cholera outbreak because it's associated with plankton. Now, zooplankton, the animal component of plankton, don't produce chlorophyll, but phytoplankton, which appear first because of the sunlight, the warming of temperature, they become abundant. The zooplankton feed on the phytoplankton. And so we could use the plankton measurement by Landsat, that early satellite. And sure enough, that's what happened. And 20-some years ago, I published a paper in the Proceedings of the National Academy of Science showing the red line are the cholera cases down on the edge of the Bay of Bengal in those villages that we have been working with for many years. The blue line is the sea surface temperature. And we have a similar figure with chlorophyll. So it turns out we could actually predict and show the correlation of these weather factors and this human disease. And with this predictive model, we could calculate the relationships between cholera, chlorophyll, and rainfall, another factor, weather factor. And for Calcutta, or Kolkata as it's known now in India, for every milligram per cubic meter increase in chlorophyll, in cubic meter of water, chlorophyll concentration, we learned that there would be about 33% increase in the number of cases of cholera in that region of Bangladesh. And if the rainfall was a millimeter per day, with every millimeter increase, there would be a 7% increase in the number of cholera cases. And this held for Madla, the village where we were doing all of our work in Bangladesh. So we now had, 20 years ago, strong evidence of the weather having a direct impact on a massive, major human pandemic disease. We went back to the data in India. A colleague, Dr. Elizabeth Whitcomb, a wonderful woman, physician who did a PhD first in India on the hydrology of India and the relationship to infection like malaria and cholera, and then did a PhD in England, and then discovered that in the library, in the British Museum, the records, these huge maps where the Brit colonials had meticulously put a dot for every cholera death, we were able to transfer all those data to the computer and to carry out analyses. And specifically, one of the discoveries was that cholera occurred in the interior of India, but only epidemically. But along the coast of India, along the coast, there would be cholera every year, spring and fall, related to the plankton correlations. And so we then were able to determine very powerfully this relationship between cholera and air temperature, and able to put that into the model to improve it. And many others have picked up on the work that we did and have shown that rainfall, flooding, how high the river will be, because when it drops in the spring in Bangladesh, it gets very turbid. And that then brings the quiescent, dormant bacteria into the water system and causes them to become then viable, to divide, to multiply, and to become the hazard. And then other factors like organic matter, et cetera. And so as a result, we now know that cholera epidemics occur in two major ways. As a major epidemic, as occurs in Delhi, which is associated, and it's periodic, it may be every 30 years or 50 years with this massive outbreak, but it's related to rainfall, weather patterns, severe weather, and coupled with the gathering for perhaps a religious festival or a special market where there'll be lots of people, crowded conditions, poor sanitation, bad weather, bang, then you'll have a big outbreak of cholera. Whereas along the coast of India and Bangladesh, you'll have cholera in the spring when the rivers are low, and then the monsoon comes, flooding, and you'll have a heavy epidemic in the fall. So now we've got a very powerful model, a mathematical model for predicting the outbreaks of this pandemic disease, cholera. And we've gone very sophisticated. Initially, we used Landsat way back in 1990, 1995. Then we moved on to a more sophisticated satellite sensing. And then we now use some of the declassified, very powerful sensors that allow us to measure water temperature, chlorophyll in a square meter of water. And that allows us to measure these parameters in the ponds from which the villagers take their drinking water and not have to use, as we did for the very early study, a 1 mile quadrant in the Bay of Bengal, just offshore of the villagers. So we've become very precise in our prediction. And we asked ourselves, could we have predicted the outbreak in Haiti that occurred in 2010 that was devastating? And so we did a retrospective analysis. We took the data that was present at that period of time, some 10 years ago, when the outbreak, the presumed original contamination by the so-called contamination by the UN troops. But in fact, what was happening, that cholera was occurring simultaneously throughout, and notice, along the coastal areas, just as we had observed in India. And we obtained samples from those areas for studying, which I'll discuss in a moment, the DNA. But we went back to the data for Haiti. We found that in 2010, they had the hottest summer in 50 years. And in the late fall of 2010, a hurricane skirted the island and they had the heaviest rainfall in 50 years. Now this begins to sound familiar, doesn't it? Very hot weather, followed by very heavy rain, and then the big outbreak occurred in late October, November in Haiti in 2010. We did another study when a Hurricane Matthew in 2015 went through Haiti and along the coast of the US. And we took the data from that hurricane and the parameters to make our prediction of where we felt that 2015 hurricane would leave conditions suitable for cholera on the left-hand side. And on the right-hand side, you see the red is where the actual cholera took place. So we were able to use our model retrospectively to predict cholera in Haiti. And then we did some real-time prediction of cholera in Yemen. In 2017, a huge cholera epidemic erupted in Yemen. They were undergoing civil war, migration of populations from the west to the east, from the east to the west. Conditions were terrible. We put all of that into the computer model, and on the left, you can see where we predicted cholera would occur. And you can see in the bottom right, that's exactly where the heavy outbreaks did occur. We published that as a squib, a short note, in 2017, September, and it was picked up by Scientific American. A little paragraph was written about predicting cholera using satellite sensing. And a colleague, Fergus McBean, working for the British Aid Agency in England, called us in January of 2018 and said, hey, why don't we work together? You predict where cholera is going to be in Yemen, and we will locate physicians and medical supplies there. And we will be preemptively prepared for cholera. And that's what we did. And in 2018, cholera numbers dropped precipitously. And now we provide on a regular basis to the United Nations, to UNICEF and the British Aid Agency. Every month, we predict 4 weeks ahead of time the hotspots for cholera in Yemen. And they're able then to locate very effectively the healthcare personnel, safe water, bottled water, whatever, and the treatments needed for cholera, and are able to keep it under control. I'm going to switch very briefly to the sequencing, the DNA work we're doing, and it does come together, and it does impinge on using all that we are learning and continue to learn about cholera to COVID. So stay with me. What we have learned to do about 12 or 15 years ago was to extract DNA from any sample: water, food, sputum, feces, urine, blood samples, whatever. And this DNA then is sequenced, and we can use any device. It can be Illumina or any of the sequencing machines that are available. And we've built a huge library, very carefully curated, with about 160,000 genomes, sequences of bacteria, viruses, fungus, and parasites. And this allows us then to match up and identify everything: bacteria, viruses, fungus, and parasites, and also the genes that they carry. So we can determine the presence of antibiotic resistance, pathogenicity properties, and that allows us to identify everything in that sample down to species and strain. As a matter of fact, strain is important because, for example, we all have E. coli in our gut, but if you have strain H70157, you get very sick. So knowing that strain is present is critical. So we have done a lot of work. In fact, I formed a company because I felt that this was really important to do for medicine. And I started the company in 2007, and it now does very well. But what we do is we analyze all the microorganisms present. For example, in this big study, which I really need to write up, was done in Calcutta, where they sent us in 3 stages, phase 1, 2, 3, samples where they knew the causative agent, samples where they didn't know. This is from victims coming into the cholera hospital, didn't know what they had, and they couldn't find Vibrio cholerae. And then some volunteers from the hospital provided samples. They sent us just the DNA. They did all the work on culturing and testing and so forth, which took weeks and weeks. Sent us the DNA, which we're able to analyze in a day. And we found that, first of all, very interesting, the pink area, this area, are the infective agents. And we found that with a known disease, where they could say, okay, we know that cholera was present, we could detect Vibrio cholerae, but we also detected other pathogens. So it turns out that infections are really polymicrobial. More than one infectious agent can be acting to cause the disease. When they could not determine what was present, we found that it was a mix of pathogens: Salmonella, Shigella, enteropathogenic E. coli. But what was fascinating was their controls also carried, they were healthy, but they carried small numbers of pathogens. And then we downloaded from the NIH database the microbiome data for healthy Westerners. And we do, some of us do, there's a very slim pink line. Some of us do carry a few pathogens. We're perfectly healthy. But the other thing that was fascinating is that this blue area represents the kind of bacteria that you get from eating yogurt and drinking buttermilk or fermented milks. We Westerners don't eat that much yogurt, but Indians, for every meal, yogurt is used. And so they're protective. And that may be a custom that has developed because of its medical value or public health value. The other thing that we discovered was that if you read across, these are the various antibiotics. We tested and we found that in the the samples from cases where they could not diagnose Vibrio cholerae, they all had antibiotic resistance. And even the healthy people, because in India, you can buy antibiotics without prescription, even from peddlers on the street. So there's a tendency to overuse antibiotics, which means that the gut flora shows that there's very high antibiotic resistance present. Now, one of the other factors that's really fascinating is that we've gathered data from about 600 studies from about 50 countries. And we found that you can use this gut flora, the kinds of bacteria that are in your gut, to characterize you as an American or as a Chinese person or a Spaniard or whatever. And it can be used as a bioforensic tool. So if you come to the US and say, well, I've always lived here, and we do a gut analysis, we could tell pretty much where you came from, which I think is fascinating. We've done a lot of interesting work on Crohn's disease, doing a big study with a colleague at Harvard and Mass General, where we're looking at men and women who suffer from this intestinal affliction. And we found that the green are the kinds of good bacteria that are present in healthy individuals, and red are the kinds of bacteria that are very dominant in those with Crohn's disease. So it's a good diagnostic. And we've done similar work with celiac disease, studying babies when they are born. This study with Dr. Fasano includes about 300 babies. Parents' genomes are sequenced, the babies' genomes are sequenced. Those that have genes for susceptibility to celiac disease, and those who have no genes for susceptibility. We are studying this and we're finding it's very complicated, but again, we can determine a difference in the kinds of bacteria in the healthy individuals that have no propensity to the disease compared to those who do carry genes for the disease or who have symptoms of it. And specific bacteria are present in the afflicted, but not in the healthy. So what is all this? And we can determine the difference by a variety of statistical techniques, healthy individuals and Crohn's disease individuals. So now I finally come to COVID. What does all this have to do with COVID? Well, it turns out that, as we all know, because after 2 years of reading the newspapers and having friends become ill, and unfortunately losing colleagues and friends and family as well, tragically, we know that this virus, now this is a virus, it's not a bacterium. Cholera is caused by a bacterium. This is a virus. And this virus attaches to receptors of a cell that has a little site that it links to in the lungs predominantly for the original variant of COVID. The Omicron seems to have greater propensity for the upper respiratory tract cells. But in any case, this ACE2 receptor is present in lungs, liver, kidneys, in various heart and blood vessels, and also in the brain. And so we have this mix of symptoms because the virus is able to attach to invade cells in various parts of the body. We were able very early on to identify the virus in a stool sample with no difficulty. We were able to pick up virus COVID-2 and its variants. This was work we did back in March 2020, almost 2 years ago. We started the work, and we very early on, this is not our work, but other scientists showed that at least 50% of those who get COVID will excrete it in the stool. These are the individual volunteers, and then it shows the number of those who carried in the stool. What's very important, now this looks like a very complicated slide, but it's not. These are patients, 1, 2, 3, 4, 5, 6, all the way down, 41 patients. And they've all been tested. The red line is the throat swab, and red means it's positive. The orange line is a fecal sample. What's fascinating is that when the throat swab has gone negative, they're still shedding virus in the feces for maybe a month or two. So it turns out that doing studies of wastewater is important. And so we have been working with the Department of Environment in Maryland and the Department of Health. They've been terrific through the governor's office, testing wastewater plant, sampling it in different locations in Maryland and in the DC area. And here's one example. Starting in June of 2020, we were sampling in Frederick, Maryland. And we were doing very exquisite testing. In other words, we used the recommended target genes to detect it, the virus that CDC recommended, but we also used the orange, another set of genes, to be absolutely sure that we were making the right diagnosis, because this was very early. This was 2 years ago. And you can see that by July, there was a big peak in Frederick, Maryland, in the wastewater. And within a few days, cases began to appear. So this becomes a very interesting mechanism for determining the health of a community. And we continue to do these studies. In fact, we have done studies at St. Mary's University where we analyzed the wastewater from given dormitories. And we were able to show that one of the dormitories in this twice a week sampling, we picked up COVID in the wastewater. And so then the university tested everybody in that dormitory, and 10 were positive, 9 of whom were asymptomatic. They didn't even know they were carrying it. And so they were able to quarantine those 10 students and prevent having to close down the whole campus. So a very important procedure. We now have perfected this, and we now have used the model for predicting cholera. We've modified it for predicting COVID. And just this week, we published a paper. It appeared this week in the Journal of Tropical Medicine and Hygiene, where we show that dew point temperature, humidity, and air temperature that's outside the comfort zone of 17 Celsius and 24 Celsius, that's roughly, that's a zone where we're comfortable. But when it gets too cold, we go inside, and if the air is very dry, the incidence goes up. If it's very, very hot, we go inside for air conditioning. And again, if the air is not well circulated and gets very dry, then the virus can be very, very highly transmitted. So we're able then to develop a prediction algorithm. And we did this analysis way back in April last year in 2021. And we now provide, from this prediction, which we did in April and May of, 2020, 2 years ago. This was our predicted map, and this was the very first analysis using this modification of the cholera prediction. And these are the actual cases at that time in 2020. And so what we do now is my former postdoc, who's now a tenured associate professor of engineering at the University of Florida, and Dr. Jutla provides a COVID risk map that you can go to this site, COVID University of Florida hubarchives.com, and you can get the daily risk map for county by county in the United States. And this is maintained by Antar and the students. And the risk map is actually provides even a time series. We're now doing cholera prediction, turning back to cholera prediction, not only for Yemen, but also for Ethiopia. And we just now, this week, have had discussions with the UN to begin prediction for Mozambique and Senegal in Africa and the Democratic Republic of Congo. So we're able then to utilize the cholera prediction map and its modification for COVID prediction. This is the latest cholera risk map. This was I'm sorry, that was a very early one. This is a COVID risk map for June this past year. Okay, I'm going to close now. One final story. I've just described for you using DNA sequencing, mathematical modeling, satellites, sensors, very sophisticated. What about the people in Bangladesh? They're still living. These boats are homes for some of these families. And in the distance, you can see the houses where they live. They are still taking water from the river and from ponds. How to help them? Well, we hypothesized that plankton carry the bacteria. If we could just remove the plankton, we could reduce cholera, because from our lab work done at Maryland, we found that if you remove the plankton, you remove about 99% of the bacteria. And it's been known that you need about 1 million bacterial cells to have a cholera infection or a case of cholera. So with the work done in the lab, we then said, okay, if we could get a simple filter, and we tried men's t-shirt material and Chinese poplin, which is very expensive cloth, and a plastic filter, which is very expensive, but is used in Africa for another disease. And we found that sari cloth, used sari cloth, folded about 4 or 5 times, gave a very nice filter. And so we were able to get funded by NIH to do a pilot study in Bangladesh, where we trained women to be extension agents, to go out to the villages and show the mothers who are the ones who collect the water for the family, how to filter the water. And of course, it was important to show them that they would take this yard or so of sari cloth, fold it, cover the calash, and then use the covered calash to collect the water. And then when the calash is, the container is filled, then to take the filter off and rinse it and hang it in sunlight, because sunlight is a disinfecting for the whatever is on the cloth, and they can reuse it. And convincing them that this was useful was not difficult, because to the right is clear water, almost as clear as what you'd get from a tap here. To the left is the water from the pond and the river, which has things swimming in it, and explaining to the mothers, that to the women, that this is not good for your children. It only took one explanation and it was understood. We were able to reduce cholera by 50% in the test villages. And the fascinating thing is that we went back 5 years later to see if it was sustainable. Did they continue? Well, it turned out that the villages that we had not trained to filter had learned about filtering. So it was very difficult to determine the efficacy. But what we did learn was that any families that didn't filter, if they were surrounded by families who filtered, they did not get cholera. So it was what we now know is the herd effect that we hear so much about here. And so understanding these links between climate, weather systems, and human health are really, really important. And they're critical, and they remain critical. And safe water is a global challenge. And I've got many collaborators from Bangladesh, the International Center for Diarrheal Diseases Research, from the National Institute of Cholera and Enteric Diseases in Calcutta, wonderful students, postdocs, visiting scientists from the University of Maryland, and collaborators from other countries, from other universities throughout the US and in other countries as well. A shout out for Dr. Huck, who came to work with me as a student, got his PhD, went back to Bangladesh to run the labs there. I recruited him back to work here. He's now a professor at the University of Maryland, and we work together. And Dr. Jutla is responsible for the mathematical modeling, he and his students. Dr. Hassan is the brilliant molecular biologist who has been key to the work. And Sion Young, brilliant woman who did the curating of the database for identifying organisms. And I like the words of John Muir. He said, when one tugs at a single thing in nature, he, and I inserted, and she, find it hitched to the rest of the universe. I've written much of this in my book, A Lab of One's Own. And I thank you for this opportunity to talk to you because I enjoy sharing the stories, and I hope it's made some sense. Thank you.
That was just great, Rita. Thank you so very much. I think everyone here, I've just seen people applauding. It's trouble you can't hear it, but I think you probably even actually know some of the people you're seeing here. So Greg, do you want to take over? Is that how you want to work it?
Well, I'm happy for you to ask questions. So first, if people have questions, please put it into the chat. But let me say, since I admitted at the very front that this is the group that I learned the most from, that Rita, your presentation I found to be amazing. Just the creativity brought to bear, and to see the interconnections. And really the correlation between unexpected things, and then to see it all the way through to the public health implications of it was really amazing, and I'm continually impressed by the human endeavor that we do, and how you have brought to bear. So thank you for sharing that.
Thank you.
We have a question from Donna Vogel. Donna?
Great.
Thank you. I'm Donna Vogel. I'm a lapsed reproductive biologist. We try to put a Jewish spin on these discussions whenever we can. And I've been sort of racking my brain to come up with a question that can relate your very informative and thought-provoking talk to that theme. And in the Bible, there's a lot of discussion of skin diseases and other afflictions, but we don't hear a lot about diarrheal disease. Also, in the Bible, there's a distinction drawn between Egypt, which gets its water from the flooding of the Nile, and pretty much the rest of the Middle East, which depends on seasonal rainfall. So my question is there any historic data or any data about the difference in diarrheal diseases between Egypt and the rest of the Middle East?
Indeed, there's a big difference. In Japan, for example, cleanliness for them is next to godliness for sure. The incidence of diarrheal disease used to be a problem, let's say a couple hundred years ago, but with the introduction of sanitation, it's probably the safest country in the Far East to visit. And similarly, Singapore. And so the relationship between sanitation and safe drinking water is extremely high. In fact, I spend a lot of time now with Safe Water Network. It's an organization that was founded by the late Paul Newman and his wife, Joanne Woodward. And it's run as a nonprofit now. And we provide kiosks that give safe water. And we work in Ghana, and in India, and it's very successful. So I think access to safe water and sanitation are key for public health. And I wish that the Gates Foundation and other foundations would pay more attention to it. Vaccines are fine, but it's sort of, the horse is out of the barn. You want to stop it from the get-go, which is safe water and sanitation. I'm not sure that answers the question, but it's my attempt.
Are there other questions? Wow.
I think I've been overwhelming.
Oh, it was fascinating. In fact, Rita gave us a reasonably similar talk to the retired faculty at the university recently, and the response was, people found it fascinating. Do you have anything you want to talk about with regards to some of the issues Donna brought up? Greg?
I'm sorry. I was thinking about I was thinking about the presentation about the predictive aspect of it and other viruses or bacterial issues. What are other applications of using this type of modeling with other types of outbreaks?
Well, that's a good question. We've done studies with West Nile, and we were able to do and published a prediction for West Virginia. Antar was first at West Virginia, then he moved to Florida, and they had a big outbreak, and the same predictive model worked extremely well. I'm sure that it will work with malaria and just about any vector-borne disease. And I like to think of cholera really as a vector-borne disease, except the vector doesn't fly, it swims.
Right. But you're right. The heat and the water are the same types of things for the mosquitoes as well.
Exactly.
So moving beyond Bangladesh, are other countries or other, the World Health Organization or other types picking up on this and using the predictive model?
That's a great question, and the answer is yes. And I can say yes because just this past week, as I say, we were meeting via Zoom with the UN and the WHO. The new person who's just come on board, she's terrific. Who heads up the cholera component of the World Health Organization, and we're all working together now, hoping to expand this to the entire African continent because that's where the problem seems to be centered predominantly.
Yeah, and what have you observed as some of the, this is outside your expertise, but
Sure.
probably in your experience, just some of the barriers to moving this to scale, expanding it to more countries? Is it simply money? Is it
You don't laugh, but I think one of the biggest barriers I've had is being a woman. If I'd been a man, this would have been hallelujah. So I've written about that in the book, but it's very interesting. I'm sure, well, I can hypothesize that one certain scientist is kind of annoyed that he didn't make the discovery of the cholera bacterium being naturally occurring and having a dormant stage. But in any case, I've had a great life and I've been able to do a lot of traveling. So it's been fun.
absolutely?
Dennis Asquith has a question. Dennis?
Yeah, if I'm allowed, a couple of personal questions. The first one would be, how do you account for your productivity at your age when most scientists would have been retired at a much younger age? And my second question is, have you ever thought about the large number of similarities between you and Rachel Carson, beginning with the same initials? You're both fascinated by the ocean. You both live in Montgomery County, and you even look a little bit like each other.
That's
And both were at the University of Maryland because
That's
Rachel,
right.
zoology department early in her career.
And you both dealt with this glass ceiling for women scientists.
Yes, that's interesting. Well, I'll let Art Popper deal with the Rachel Carson lookalike, but let's see. The first question is I think kind of easy for me. Good genes. My dad died dancing at the age of 75 on a trip to Italy. So he was at a wedding, not his, but with my mother, and that's one. But my father and mother were immigrants, and we grew up in Beverly, Massachusetts, and Dad grew everything that the family ate. So we were on a vegetable, fruit. He even raised chickens. So we definitely went straight from the garden to the fork, if you will, to the table. And then I've always been a jogger. I've been a runner. I just walk fast now. I walk 3 or 4 miles a day. But I think that plus not being a vegetarian, but being semi-vegetarian probably, and growing up along the coast where fish was abundant, I think all of these things that we now learn are good. I had them by chance, not by selection. So plus the other thing I think is really important is that I married the right guy. I was married for 62 years to Jack Colwell, who was exactly the kindest, and he was a physicist. He and I raised 2 great daughters, and we now, Jack passed away prostate cancer 3 years ago. But we raised 2 daughters, and we have 3 grandchildren. So I've been blessed, I think, all the way around. Family, friends, colleagues, and good health. So I just wish that our grandchildren, your and grandchildren, can have a world that's safe, and I worry about that a lot.
Well, that sounds like I'm conscious of the time, and that sounds like the right note.
Elliot has been raising his hand.
Why don't let Elliot
Oh, okay.
have his hand up?
Please.
Elliot, you're muted.
Yes, just one question about the role of rainfall in cholera. Is it purely a mechanical thing that the rainwater washes the bacteria from the shoreline into the seawater?
That's very interesting. It's a combination of humidity and effect on salinity and flooding. So it's a weather phenomenon and not just mechanical. And Jay, was it you who had another question?
Yeah, just sort of a comment. With the climate change and getting much, much warmer, I'm going to assume that we're going to get these outbreaks all over the world now.
Well, you asked
I
that.
mean, even in Siberia, where the temperature has gotten up to 100.
You asked absolutely the right question, because we published a paper with colleagues in Italy, Carla Pruzzo and her team in Genoa, and a team at Max Planck and a group at the Marine Laboratory in England and us at Maryland, where we showed that the Brits had collected plankton every year for the last 50 years. And we went back and we extracted the DNA, and they had the temperature records of the North Atlantic. And what we did, we showed that there was an increase in the number of bacteria, Vibrios, and we correlated it with increase in number of cases of infections in Europe, England, and the North Atlantic of the US. And we published that 5 years ago. So yes, with climate change, we can anticipate a definite effect on our health.
By the
So
way,
Well, I think,
I'll let you close that one. Yes, go ahead.
well, Rita, thank you so very much. This is a great talk, and we really appreciate it. I will remind people her book, Rita's book, is available on Amazon. I wasn't paid to plug it, but I think Rita has a lot to say. She made a comment about how things would be accepted if she was a man instead of woman. She has a lot to say about the role of women in science. I think it's something a lot of you would find interesting. So it's
Art,
even on Kindle.
Let me check this last thing. So if I put your presentation correctly, that cholera is found in the plankton in the bay, which makes its way into shellfish, which means we should keep kosher. Is that right? Is that a takeaway from this?
Yeah.
So probably not.
I keep kosher.
Okay.
I don't eat raw.
Me too. So now thank you for being here. Really, really interesting.